Rod, and this was a question that was asked actually in the other panel, which was, microbiologically, when methane is mitigated, which microbes actually could, pick up the slack?
Sinead, thanks for the question. I think it's sort of very difficult to provide a comprehensive answer. So in terms of which particular functional group that you'd like to see enhanced when you do that methane mitigation, that would be things that are making propionate as a major target. And I think that the natural sort of dietary experiment has been done. So that's the difference between concentrate diets and forage diets. And so if you look at just the stoichiometry of that, you can see that per mole of hexose fermented, producing more propionate and butyrate greatly reduces the methane yield per mole of glucose fermented. Of course, it's not as simple as that, and the room system is very heterogeneous, and there are many other factors that play into it. But I think that the idea of having functional groups rather than which organisms or a specific name, I don't think that's not really feasible. So it's more a consortium, a group of organisms that are involved in this cooperative, collaborative type relationship of producing hydrogen and utilizing that hydrogen. And so I don't think there's a name. But, you could say, well, I think what we should be doing is looking at combinations. And I think the whole emphasis is just on methane. And I think we've missed the plot. I think it's more about interactions, and not only interactions between microbes themselves, but with the host animal and the host physiology, because that's obviously very important. But work's been done on Megasphaera elsdenii. Yeah. So there's another name, Selenomonas ruminantium, species Lactolytica. Those are obvious candidates. I think the other group that people are trying to promote and is a reductive acetogenesis. And that's just not a competitive process at all. And making an acetogen produce acetic acid from CO2 and hydrogen is really uphill. It's very steep. And it doesn't happen because of their versatility. Yeah, so maybe that's enough.
Yeah, thanks, Rod. That's awesome.
So maybe I can expand on this a little bit. Because
I
think what I want to emphasize here is really that what people really hopefully appreciate is that and I alluded on that we have new technologies available now. So with DNA sequencing, right? So Rod was talking about functional groups or organisms that can perform a certain function. We don't really know in detail what's going on in these really complex systems. But fortunately, we now have the technologies available that allow us to actually start digging into that. So I think this is really important to keep in mind. Room microbiologists have been working on trying to understand what's going on in the room for the last literally 100 years, starting with Bob Hungate. But now we actually have tools that allow us really to obtain a mechanistic understanding. And that will allow us also to redirect not only hydrogen flow, but also other components. So I think we should see this as an opportunity and really understand that we are really at an exciting time where we actually can make a difference.
Cool. Thanks, Matthias. Yeah, I think that's really interesting. When a lot of people talk about stackability, they're often talking about stacking mitigation on top of mitigation. But actually, when I think about stackability, I'm talking about mitigation plus productivity. And that's the stackability I'm wanting to say. We've got a question over here.
Hi. For the whole panel, and I wanted to kind of piggyback off of what you just said. When you think about stackability, I wanted to ask the question to our dairyman representative, Jed. What do you think the industry is interested in terms of, I believe you said that they were on board, and I want to know what exactly they were on board with, because it sounds like there's a willingness and interest to sort of play, understanding the importance of methane emissions reduction, and whether or not productivity is going to have to be part of the solution answer, or whether there are other things that are important to the dairy industry, to the cattle industry. And before you answer, I also just want to thank the whole panel for the great analogies. We've got capture the flag, we've got engines, and we had one other one, I think. Oh, the messy bedroom, thank you very much. And Sarah, I love the back of the napkin. So thank you, panel, I love this panel. And Jed, please tell us more about what the industry really would be willing to entertain or what it needs to make changes. Thanks.
So I'll speak to my clients and the ones I interact with. That's all I can speak for. So my client base is very engaged in adopting new technologies. They want to try new things. They're not opposed to it. They're committed to being a part of the dairy industry for their lives. Most of them are second generation, some are third, and this is not going away for them. They are committed to being a part of this, and their kids are part of it. And so they're committed to the state's goals, their co-op's goals, and so forth, as what that means as it relates to methane. When we talk about the cost of methane mitigation, the carbon markets, and so forth, if someone brought a product to market that would, its carbon transaction offset the cost of the product, that's a barrier to entry that's gone at that point. Consumption of that is a non-starter. We can do that tomorrow, and we have, and that's where AgNext came in. Myself and a dairyman sat down with a creamery who had no interest in heading this direction, but we minimized the cost ourselves and did the math, and it worked. And so we just started that process on his own because he wanted to know what else he could do with the goal of keeping his market for his milk open. And so that's their mindset. From a productivity perspective, it's really hard to come into a very efficient system and add more efficiency. That's what they're doing every day. And so economically, I think if we can offset the cost of these products, the barrier to entry is non-existent. And then as we work towards that, there's more and more opportunities as we go through that process.
Thanks. Do we have another question? Yeah, down here.
Yes, I have a question, and actually, Jed, you're on the hot spot here because you brought it up. You mentioned RBST as a case study of something that improved efficiency and got taken off the market. I guess my question to the whole panel would be, how do we stop that from, since we're talking about fiction, how do we stop that from happening to feed additives?
I guess I can go first on that one, since I said it. Consumers have to be educated, they have to be engaged, and we have to get their buy-in before we start. If we surprise them with concepts they're afraid of, they're just gonna balk and say no. And we've been down that path. The industry lost because of it. Our efficiency's lost. From an intensity perspective, we went backwards. Now, as we talked about genetics earlier, we've done great things on the front of genetics and really improved these cattle. Joe McFadden mentioned some component data from the East Coast earlier, and 10 years ago that was unrealistic, and now it's commonplace. And so we just have to bring the consumer with us on this and get ahead of that part, or it'll blow up in our face.
And I think this is also even more important for technologies like CRISPR, right? So I think one of the things that we're working on with Ted Audacious is really trying to engineer the microbiome. But we have an entire group dedicated really to outreach, to trying to educate people on what we are doing, so that they understand really that there should be no safety concern, or that we actually try to have several roadblocks or safety measurements in place before we actually apply any of these technologies into an animal or even bring it into market. So I think, Alison, this is a great question, which we're aware of, but fortunately we also have the resources to bring in those people to really help us to bring that message and be really transparent to what we are doing in the lab, and not suddenly just like creating this monster that we just basically put out there. And I mean, that has happened in Europe. When I grew up, there was this case with Monsanto, where basically entire Europe was kind of rejecting then genetics, right? So I think we have to be aware of that and be proactive.
Cool, thank you. Do we have another question from the audience? If not, we do, over here, yeah. Gee,
Hey,
I was just about to ask one.
I'll try to keep it quick. So yeah, thank you. There's a fair amount of literature about other organisms that will eat a diet similar to a cow, like kangaroos and some termites and so on, and a lot of interest in poking around to prospect for solutions. Can you assess before we do that if there is some big picture idea of whether those systems are net anywhere near as productive as a ruminant at conversion? Like, are we going to go down that road and find some microbe or some enzyme and then realize the whole thing was only 50% as efficient as a cow after all? How do we know if that's a route we should go down?
So let me take a stab at that. I think that there are multiple systems. Just as a background, the ruminant system, that's the most highly evolved and probably the most recent development for mutualistic gut fermentation. And that's because most of those things like forage and even concentrates end up in volatile fatty acids, and the volatile fatty acids contribute 70 to 80% of the metabolizable energy requirements of the animal. And so that's very different to most of these other systems. So the ruminant physiology is really key to the whole thing. And I'm not saying that you couldn't go and look in termites and find lots of treponemes, things like that, that are involved in probably intermediate steps in the process of carbohydrate degradation. But whether they're going to be a good source of solutions to what's happening in the rumen, I'm a bit skeptical. Kangaroos are now, that's really an interesting, it has foregut fermentation, but it's not a ruminant. So it doesn't ruminate and it doesn't regurgitate this gas. It doesn't have the same gas production potential. So these are very, very different systems. And I'd be surprised if you found something in those systems that could really be a game changer in the rumen. So I think they're interesting biologically, and they're fascinating. And so maybe another kind of analogy would be looking in goose shit for fiber digesters. Well, no, they eat fiber, yeah, but they just extract solubles, and the rest is what we find on the ground. Those are the landmines. So and there's a lot of it. So just because they're eating something doesn't mean they really digest it. So it's the equid versus the bovid kind of competition and how that evolved.
Thanks, Rod. Did you?
And
How bullshit replaced horseshit.
And you might find an organism that does one reaction really much more efficiently, right? But the challenge you will have is you have to take that microbe or that enzyme and then have it establish itself in an already established community. And so the challenge to do this with a non-native organism will be much higher than for an organism that has already been established before.
Thanks. So I'll do one more question, and it's to all the panel members, I think. I'd like to get an answer from you guys on this. So we've hypothesized that the energy transfer, about this energy transfer for nearly 60 years or 100 years, depending on your date, your timeline. And I think Sara gave some thoughts on why it's been so difficult to demonstrate that this energy transfer is real. I think those animal numbers for those trials are eye-watering and expensive. But do we actually have any evidence to demonstrate that this will be possible? State of play right now. Asking the panel. Yeah, quick thoughts. Yeah, who wants to go first? And then you can be released.
I think people that have studied the system and know a lot more about energetics than I do, I've worked out that the amount of methane not formed versus the amount of energy you lose as hydrogen lost. So that's why we don't want to get beyond 20-30% methane reduction is very, very little, and it's almost a wash. And so you're not really saving a whole lot because you're losing that hydrogen. So the whole thing is redirecting and capturing hydrogen. But I think that the idea of this big range of gross energy is kind of a little bit misleading. And if you convert that back to something like metabolizable energy or net energy, it's even less. And there's not a lot of gain there.
So to summary, lower our expectations?
Well, I think so. I think we need to operate in a range where you still have full, rumen fermentation at a rate where you don't impact dry matter intake, so you can't drop dry matter intake, but still have methane reduction without changing productivity. That would be optimistic.
Okay, thanks. Roderick.
That's the best.
Yeah, Jed?
All I can say is from the field right now and feeding on the 4 herds we have not seen any losses or gains. And so I think it's a neutral point.
Yeah. Well, thanks. Matthias?
I think your question was if we are thinking it's possible, and I think short answer is yes, I think it's possible. But I also agree that we might have to lower expectations out the amount that we can lower.
Okay, thank you. And Sarah?
Yes, that's back to the energy of it. It's not that much energy, right? From a standpoint of the point was brought up well from Rod of once you discount everything, it's that 2 to 12% becomes a lot smaller, right? In terms of what can actually be turned into net energy for gain or lactation.
Cool. So with that, I'd just like to thank you guys for listening, but also to thank our expert panel as well. Thank you.